region where all those potassium ions are, and the outside of the cell is where that sodium ions are. So this gradient is created by those sodium and potassium ions. When the neuron is at rest, there's something called resting potential that occurs. And in that case, there are more sodium ions on the outside than potassium ions on the inside. So there's more positive charge outside, which means the inside of neuron is what charged? Someone tell me. Oh, Arya? Negative. Yes, negatively charged. The neuron is negatively charged on the inside of the cell. Ions cannot pass freely through this membrane, so that is how that connection is maintained. If the ions could pass freely, then obviously the sodium ions would move inside the cell and try and make sure that charge was balanced. But because they cannot pass freely, that is how that charge is maintained. So neuron is allowed to be at rest by those leakage channels. So if you see those blue channels over there, those are sodium leakage channels. And the potassium, the pink ones are the potassium gated channels. So these basically allow more potassium ions out of the cell, because as you can see, there's a lot more potassium gates and potassium channels than there are sodium ones. So they allow more potassium to go outside the cell than in allowing sodium into the cell. So again, that also works to maintain the charge where the outside of the cell is more positively charged than the inside. Okay. So you have something called a stimulus, and this is what gives the opportunity to change that electrochemical gradient. So when you get neurochemical signals from other neurons or any other sort of type of stimulus, for example, touching something or doing something, that changes the voltage inside the cell. And it changes the potential across the membrane because it might introduce more positive charge inside the neuron. If the change is very small, nothing really happens. Those leakage channels will basically work to help revert the gradient back to normal, back to that resting potential. So that's something called a graded potential. A graded potential is when the stimulus isn't strong enough to actually affect the neuron long term. So if you can see on the graph there, you'll see there'll be like a slight increase, and then it'll go back down, a slight increase and back down. So that change will be reverted by the leakage channels. But if the change is high enough, and the neuron's the internal charge increases enough, it can start a chain reaction, which is called an action potential. So let's get into action potentials. Once the difference crosses a certain threshold, an action potential will begin. This is driven by the voltage-activated gates that line the cell body and axons. So they're similar to the leakage channels, but instead of being constantly open, voltage-activated gates only open when a certain voltage is reached, hence the name voltage-activated gates. So first, when a certain threshold of voltage is reached, the sodium voltage-activated gates open, and they allow a flood of sodium ions to move through the membrane into the cell. This introduces a spike in positive charge because there's suddenly so many more positive ions inside the cell. And this increasingly positive charge then opens other voltage-activated gates further down the axon. So as you can see, the positive charge moves through the axon because it opens more gates. And then those gates that are further down the axon let in more sodium ions, which increases the charge, which opens the gates further down. And that process continues, and you have a action potential propagating down the cell. And so then this is actually called an action potential or a spike or a neuron firing. There's a bunch of different words for it. And then later, when peak action potential is reached, the sodium activated gates will also open, allowing potassium to leave the cell, thus by reverting back to a more normal gradient and reducing that positive charge. So you can see that positive charge only travels through at a certain interval, but once it passes, then the potassium gates open and allow the cell to revert back to a more normal gradient balance. Okay. Oh, you can't see the video. Oh, you can't see the video. Okay. I just realized I forgot to do the audio thing, so I'm not sure if this will work. If it does not, then it will give me 3 seconds to make sure it works. Nope, it's not working. Okay, give me 3 seconds, guys. I want to make sure you hear Hank Green. So this is basically going to be a video that will help us. Hi, Arya. That will help us learn more and visualize more about what action potentials look like.
Did you put this or something?
Hmm?
Did you put this or something?
is an action potential. And your best bet for making
But
that
it's coming
happen
from
is
my
to
laptop.
depolarize that resting neuron.
Give me 3 seconds.
the mic down.
No, I can do it. Right. Just give me 3 seconds. Okay, we are going to do it this way.
I mean,
Oh.
cause a big enough change in its
Can everyone
membrane
hear the
potential
audio?
that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is gated channels. That is an action potential.
Sorry,
And your
let me
best
start
bet
over.
for making that happen is to depolarize that resting neuron. Cause open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all or nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them. And that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level.
Okay, awesome. So that was a visualization of action potentials. So again, this is more images of just what that looks like and how that electrical current moves through. And now we're going to take a closer look at the actual numbers. So first we have resting potential, that's at the numbers 5 and 5 on either side. And then we have the threshold value. That's the value that actually causes action potential, and that happens at around negative 55 millivolts. Please remember these numbers, they're very important. And then from numbers 2 to 3, you have depolarization, is where you reach that highest voltage, that peak, which is around 30 millivolts. And then after that peak down to about where 4 is on this graph, you have hyperpolarization, which is that fall where the sodium or the potassium activated channels open and basically bring the charge back down, and it brings it down too far, as Hank Green said, and brings it down to around negative 70 millivolts. And then you have repolarization, where you return back to rest at, again, around negative 70 millivolts. So does everyone understand that? Are there any questions here? Because this is like the fundamentals of neuroscience. I want to make sure you guys understand this. Okay, perfect. So basically what that means is every thought or every action you've ever done is simply a combination of action potentials. So this is an exercise my neuroscience teacher did. So lift your hand and give someone around you a high five. High five, right here. That, action potentials. And another one, he said, imagine an elephant in a pink tutu balancing on a red circus ball. I want everyone to close their eyes and imagine. That is also 100% just action potentials right there. Neurons firing and neuron transmitters being sent through the brain. Okay, so why is this relevant? Why is this actually important for us to learn? So if I give you a very loose description of human learning, it's neurons firing and sending information to each other, and every time they send that information, they basically take information and might change it and manipulate it slightly before they send something slightly different than what they receive to the next neuron, etc., etc. And between connections and synapses, these connections are strengthened or weakened depending on use. So the brain does something called pruning, where basically if you use a connection between your two neurons excessively, you use it a lot, that connection is really strengthened. They grow more branches, and the brain basically makes it so that connection is stronger. Whereas if you have a connection that you use less frequently, the brain will make it less strong, will basically remove branches and make it weaker, to the point where it might actually remove that connection entirely. So what the brain is doing there is trying to make optimal connections and do optimal calculations and sort of transformations with the data neurons are getting in order to make the thoughts in your brain. So this actually inspires neural networks, which we're going to learn about next week. In neural networks, the goal is to have the nodes or the neurons take incoming information, apply mathematical transformations, and then pass that data on to other nodes, similarly to how a neuron does it, or very loosely similarly. And then for features, the goal of those features is to emphasize and weaken features in the data that will help the model predict something. So the significance of some features are reduced and the significance of others are increased, similarly to how the connections and synapses work. So basically what I'm trying to get at is what we're learning about here in the brain is sort of what inspires very loosely neural networks. So you can see that web of neurons, that illustration of neurons up there, and what is going to be a very familiar image to you as we get into the deep learning neural networks next week. Okay. So I want everyone to talk to the people around you with the very minimal information you have, or maybe any learning you have yourselves. I want you to answer two questions. What is one way neural networks are like the brain? And if you have enough information to answer that question, please, if you have more information, please get into it. And what is one way that they are not? TAs, again, please facilitate your group discussions. Arya is not here, but I will be going around. Awesome. Everyone turn around and talk to your friends.
They both have functionally or based on the use case. I know a little bit about it. Certain goals, neural network has. I really don't know much about them. Yeah, it makes sense. Try to simulate the brain. Yeah, like we got both use signals.
A good answer. I heard that one. I like it. Yeah, they've already been discussing. Did you guys name one way the neural networks are not like the brain? He gave you an answer, but I don't think either of you heard him.
Well, I mean, neural networks are intangible. They're not like based on chemicals or anything.
That's a good one. You're just giving his answer? No, come on, come up with your own answer. I believe in you. You don't know any? Well, for one, neural networks have only one layer. So they have like different, like if I go back to the image here, you can see it's a system of layers like that. Whereas like the brain is just like a random web of neurons. Every neuron can be connected to like a billion other neurons. Whereas neural networks are a lot more organized. Yes. I got you, don't worry. Okay, I'm gonna give you one more minute to wrap up whatever you're saying. Okay, perfect. Let's move on. So now we're going to get into the actual brain and the different parts of the brain. So the brain is split into different regions and lobes. Each lobe has a specific function and a purpose. So here are some of the examples of the regions we're going to be covering. Does anyone know anything about any of these regions? Please raise your hand. I believe in you. So, we got one right there. What do you know about any of these regions? Yes, the frontal lobe is for thinking, critical thinking. Yes, very much correct. Anybody else? I'm going to take at least 2 more before I move on. Oh, yes, Ananya?
The brainstem consists of the thalamus, which is important for relaying information to your central nervous system.
Yes.
And also very important for like things that you don't necessarily like control with your mind.
Mm-hmm.
Or eating and stuff like
Yeah, so the brainstem is for all primitive functions. We're going to go over that in a second. So like involuntary things, that's a good one. One more. I believe in you guys. Oh, yes.
Pretty sure the cerebellum is for like coordinating muscle movement and motor control.
That's a good one. Okay, perfect. We're going to go into all of these details in a second, so don't worry if you don't know anything quite yet. Okay, so the first thing we're going to get into is the frontal lobe, which is, someone said over here very accurately, is for thinking, critical thinking skills, decision making, your personality is very much housed here, planning, impulse control, reasoning, and voluntary movements. So this is the part of the brain that a lot of people say develops last because it's a lot of those like really complex critical thinking skills that are quite unique to human beings. So the main parts of this are the prefrontal cortex, which is involved in complex behaviors such as decision making, and also that personality is really housed there. You have the motor cortex, which controls some of the voluntary movements and motor functions. And then lastly, you have Broca's area, which is a very important for speech production and language processing, typically in the left hemisphere of the brain, I believe. Second, we have the parietal lobe. So the functionality of this is to process sensory information, things like things you feel, things you smell, things you see, things like that, spatial awareness, and also navigation. So it basically assembles and processes the senses and like whatever sensory information you get into information that the brain can actually understand and process. So for example, if anyone knows that like when you see images, they're actually flipped upside down in your retina. So this part is the part of the brain that will flip them back around and make it into something you can actually understand. So the main parts of this are the somatosensory cortex, which processes that sensory input from the brain, including touch, temperature, and pain. You have the angular gyrus, which is involved in language processing and also number processing. And then the supramarginal gyrus, which plays a role in language perception and also that spatial awareness that I was talking about earlier. Then you have the occipital lobe. This is the main vision center. So the functionality is, of course, that visual processing and recognizing those shapes and colors and movements. Also, depth perception is hugely important. That is all housed in this part of the brain. So the parts of the brain you have in this lobe are the primary visual cortex, which is the main area for processing visual information. This is actually what inspires computer vision, which we're going to be learning about in 2 weeks right after deep learning, so remember that part of the brain. And then you have the visual association areas that are involved in like the higher level processing of visual stimuli, including like color, motion, object recognition, recognition of different like things that you've seen before, familiar objects, etc. Then we have the temporal lobe. The temporal lobe is where auditory processing happens. Memory is housed here. More language processing. Language processing is in several parts of the brain because it's so important for, human growth. And then you also have facial recognition. So where you recognize faces, that all happens in this part of the brain. The main parts of this are the auditory cortex, which processes auditory information from the ear. Wernicke's area, which is critical for that language comprehension. Again, usually in the left hemisphere. This is really used to understand spoken language, whereas other parts of the brain might be used for written language, etc. Wernicke's area is very specific for spoken language. Then you have the hippocampus, which is involved in memory formation and also spatial navigation. And then also the amygdala, which is involved in emotional processing and like processing of emotions, particularly like the heightened emotions like fear or pleasure, things like that. Okay, then we have the cerebellum. This also coordinates voluntary movements, controls balance and posture, and is also responsible for like really fine-tuning those motor actions. So there's really like small movements and delicate movements you can make with your fingers or any other parts of your body, that all comes from there. And it's also involved in motor learning and motor memory. So like muscle memory, when things are in your muscle memory, that's all part of this part of the brain. So the two parts of this brain are the cerebellar cortex, which is that outer layer of this region that really processes that sensory information in order to coordinate that motor activity. And then you have the deep cerebral nuclei, which is involved in relaying information to other parts of the brain and also facilitating that movement control, that like fine movement control. Okay, and then we have the brainstem, which was also brought up earlier. So this really controls those basic life functions, those involuntary autonomic movements like breathing, heart rate, blood pressure, digestion, etc. And it's a pathway for signals from the spinal cord, by extension the entire body, because all the body's sensory information goes through that spinal cord up into the brain, and the brainstem really serves as a way to take that information and take it where it needs to go in the brain. It also regulates alertness and sleep. So the parts of this brain are first the midbrain, that really connects the brain to the spinal cord and regulates those visual and auditory reflexes. So sort of the information that doesn't have to go all the way up to your brain before an action is done. Like when you're touching a hot stove, you're not really sitting there and thinking about the fact that like, ow, I'm touching a hot stove. It happens instantaneously, and that is all due to the brainstem and the midbrain specifically. And then you have the pons, which relays signals between different parts of the brain and is involved in regulating that sleep and also that arousal sort of information and different things like that. And then you have the medulla oblongata, which controls autonomic functions such as heart rate, respiration, etc. Okay, and then you have some other important functions, such as the thalamus, which, as Ananya said, is right above the brainstem there. And this sort of sorts that sensory information. It basically deals with sort of understanding like where the information needs to go. However, one important fact is the thalamus does not deal with smell. Smell operates through a different channel in the brain. And it also contributes to that sleep and alertness regulation that I discussed earlier with the brainstem. And then you have the hypothalamus, which hypothalamus is right below the thalamus, and that really controls homeostasis, regulating the body's state, regulating temperature, driving hunger and thirst to get the body the nutrients it needs, and that sleep-wake cycle, etc. So that really, and also hormone regulation and emotional responses are also driven by this area.